Thank you. You should have
Yeah,
reminded me earlier. All right,
sorry.
sorry. The first part of this lesson wasn't recorded. We're recording from here on if you didn't see this lesson. Uh, I don't think I'm sharing now. Yeah, once I start the share, I don't see that. So, uh, another thing, once I'm sharing, if somebody puts in a comment, I won't see the comment. So that's why I don't use the comments, because I like to share my screen.
That's okay. Would you just be able to post these slides when you get a chance?
Yeah, I'll post the slides. If you remind me during the lab, I'll post them during the lab. Okay?
Perfect. Thank you.
All right. All right. Any questions about Chapter 8? If not, let's
No.
end Chapter 8. End Chapter 8 and begin Chapter 14, I think. It looks like somebody's adding late. If you're adding late, I just recently turned on the recording of this lesson, so you have to look heavily on the slides because the early part of this lesson wasn't recorded. All right, chapter 14. Like always, I have an introductory slide showing you the major goals and a rough outline of what we'll be talking about. In this chapter, you do need to know the terms. I've only got 3 listed here: pathology, etiology, and microbial antagonism. Actually, this chapter, about 50% of it is the terms. So if you learn the terms to this chapter, you've learned about 50% of the material to this chapter. So this chapter is a lot on terms. They're not difficult. They're, what causes disease and why we get disease and how disease spreads, things like that. Anyways, understand the relationship of the normal microbiota and its host. We'll be talking a little bit more about that in detail today. Understand the stages of disease development. Every disease starts with the initial infection and then goes through different stages which give us disease. And then lastly, understand what nosocomial infections are and how to control them. And let me briefly state that nosocomial infections are one of the most important topics that you can learn in this class, because the majority of you are going to go out there and work in the health field, and you need to know what nosocomial infections are and how to control them, because we do not want nosocomial infections. Okay? And we'll talk more about that when we get to it. Any question about what we're doing? So here's 3 of the early terms: pathology, the study of disease; etiology, the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. Pathogenesis, the development of a disease. Any question about any of that? This is the start of the worksheet, but I decided I'm going to go through it because people seem to be having a little bit of trouble with the in-class assignment, meaning the worksheet and the lecture. So infection, the colonization of the body by pathogens, or the presence of a microbe where it's not normally found. An example of that is obviously when you get an infection of your body by getting exposed to the cold. The virus come and colonize your cells. The presence of microbes where they're not normally found. We find E. coli in our intestines, but if you have E. coli growing in your tissues, like subcutaneously under your skin, that would be an infection. Disease, an abnormal state in which the body is not functioning normally. You should understand that few microorganisms are pathogenic. Remember, we discussed pathogenic. That's an organism that is capable of causing disease. Few bacteria or microorganisms are pathogenic. Some microorganisms are actually beneficial. Some benefit the host, like E. coli helps us digest food. Many of the beneficial bacteria are the bacteria that are normally found in and on our body, and we call them our normal microbiota. The normal microbiota and their relationship with the host is the normal microbiota, or large communities of microbes found in association with all animals and plants. When usually we use the term normal microbiota, we're talking about the normal microbes that live in and on a human. Okay? And each species has their normal microbiota. Like for us, Staphylococcus epidermidis and Micrococcus luteus is normal microbiota on our skin. E. coli, I don't know if you can see, I'm pointing to my belly. Normal microbiota in our gut. But different species have different normal microbiota. And as you may know, Salmonella is a normal microbiota for chickens and turkey and many other birds. And that organism causes disease in us. So it is not a part of our normal microbiota. Any question about that? Each species has its own normal microbiota. Bartonella henselae is a normal microbiota that is found on cats, and that's the bacteria that causes cat scratch fever. It's not a part of our humans' normal microbiota. The normal microbiota are microbes that permanently colonize a host, like permanently colonize a person. The normal microbiota are only found in certain regions of the body. They are not normally found in other regions, like Staphylococcus epidermidis, to my knowledge, is only found on the skin and maybe in the what do you call that, the oral mucous membranes, but mainly on the skin. It is not found in the intestines. At least I don't think it is. Any question about that? Disruption of the normal microbiota and the host balance appears to be associated with diseases and impaired immune function. Has anyone ever taken antibiotics and you got diarrhea, an upset stomach, or constipation after taking antibiotics?
I haven't, but I've had a lot of patients who have. They get like C. diff or something like that.
Yeah, C. diff is something else. I'm talking about just a little bit off, where they got diarrhea or constipation. Anyways, that's happened to me occasionally, and it happens to a lot of patients. It's just one of the side reactions of taking the antibiotics. What's happening is the antibiotic is disrupting the patient's normal microbiota, and then the patient becomes sick. Diarrhea usually doesn't last too long, or constipation, once again, it usually doesn't last too long. And then the normal microbiota will reestablish itself, if not right away, It will eventually reestablish itself when the patient stops taking the antibiotics. On most patients, it actually reestablishes itself in less than a day. Like when I get diarrhea from taking antibiotics, it's usually one time and I have diarrhea for maybe a half an hour or an hour. And it's just a disruption of the normal microbiota and the host balance. The normal microbiota are only found in certain places of the human body, and this picture is trying to show you, such as the skin, the orifices open to the body, especially the mucous membranes, the gastrointestinal tract, the lower urinary and reproductive systems like the urethra. That's where we find the normal microbiota. And obviously the mouth, nose, stuff like that. Eyes, which that's totally open. The normal microbiota generally colonizes the patient for the life of that patient. Like shortly after I was born, I got Staphylococcus epidermidis on my skin, probably from the doctor or my parents who handled me. I mean, that's normal. You're not born with Staphylococcus epidermidis. Unless you pick it up during birth, it's possible you could pick up Staphylococcus epidermidis during birth, but it's picked up usually from your parents, and it normally is with the patient for the life of that patient. That's what the normal microbiota is. So for a very long time. That's different from a transient microbiota, which could be present for days, weeks, or months, but it's not the normal microbiota. It's only transient. I once had an example of a transient microbiota, and I went and saw my aunt, and she had a Staphylococcus aureus skin infection, and she was being treated for it, but she, how do I say this? She didn't realize that you have to be under the treatment for a while before it won't spread. And she insisted on hugging me when I got to visit her. And she transferred Staphylococcus aureus over to me. And how I knew was I was teaching microbiology, and I routinely, once in a lab, would swab my skin just to look to see if I had Staphylococcus aureus. And normally, all the other times, I never got Staphylococcus aureus. And shortly after she hugged me, I had Staphylococcus aureus on my skin. That was something we could do if we were in the lab, but you can't obviously do that while we're doing this online. Fortunately, for me, it was only transient. It didn't become a part of my normal microbiota. And the next time I sampled my skin, which was a few months later, I didn't have any Staphylococcus aureus. So it was transferred to me from my aunt, and it became a transient microbiota, but it eventually went away. My normal microbiota competed with it and overtook it, and so the Staphylococcus aureus went away. And I never saw it again, and I tested several, well, how many? About 4 times a year, and I've tested now for several years, and I've never seen it except for that one time. So that was a transient microbiota. Staphylococcus aureus, if you don't know, is a human pathogen. It can cause skin lesions, and on my aunt it did. That's why she was receiving treatment for it. When it was a transient microbiota for me, it didn't cause any skin lesions, and it then went away. The normal microbiota and the host, let's talk a little bit more about that topic. Humans have about 1 times 10 to the 13th human body cells. And we humans have about 1 times 10 to the 14th bacteria cells as part of our normal microbiota. So we have many more, about 10 times more, bacteria cells with us than we have our own cells. And that begs to ask the question, are we more microbe than we are human? Don't let the answer freak you out too much. Any question about any of that?
So as far as like a normal human microbiota that we're talking about, I've heard that if you're a healthcare worker, like MRSA becomes a normal part of your microbiota. Is that true?
No, it is not a normal part of your microbiota, but you could be exposed to MRSA, MRSA, or multi-drug resistant Staphylococcus aureus, in which case, if it causes disease in you, they will then treat it to get it removed from you. Okay? But I don't know if they ever treat it on someone if it's not causing disease. Because many people would have it and not realize that they have it because it's not causing disease. But generally, MRSA is not a part of somebody's normal microbiota. All it is a variant of Staphylococcus aureus that is resistant to many drugs and many antibiotics. And for me, that would have been a transient microbe because Staphylococcus aureus on my skin was a transient microbe. All right? Good question, but no, it doesn't normally become a part of your normal microbiota. And like I said, if it were to cause disease, they will remove it from your transient microbiota. They'll remove it from you.
Okay, thank you.
What else did I want to say about that? Don't remember. Here's a few pictures of the normal microbiota. Some the orange cocci in the nasal epithelium. There's some rods in the lining of your stomach. I think these are smaller rods down here, but I'm not positive. Those are big rods. And then both rods and cocci in your large intestine. And then maybe right there, that would be a coccobacillus. The human microbiota have been found in some surprising places recently, although this is now, 7 years ago? So not quite so recent, but still fairly recent. They found that normal human breast tissue, at least in females, and their mother's milk is not sterile. It has a normal microbiota, that there will be bacteria growing in a human female breast and in the mother's milk. The placenta harbors a normal microbiota. And I thought there was a third one. Where did I miss it? Nope, I went over it too fast. Females, in their bladder, there is a normal microbiota, so that female urine is never sterile, because there's a normal microbiota in the female bladder. I don't know if because males have a longer urethra, or if they simply didn't test males, but this study is only on females. All right, any question about the normal microbiota? So the normal microbiota is important in helping humans maintain their health. And the first thing is the normal microbiota can antagonize or undergo antagonism with the human pathogen, where the two can compete with each other. The reason why I lost Staphylococcus aureus on my skin was my normal microbiota competed with it and outgrew it, and then the Staphylococcus aureus is now gone from my skin. That's what I wanted to talk about. I was talking about Staphylococcus aureus, and I said, what else did I want to say about that? In humans, about 20% of Americans have Staphylococcus aureus as a normal part of their normal microbiota. And these individuals are much more likely to get a skin lesion than individuals who do not have Staphylococcus aureus as a part of their normal skin microbiota. And in humans, the Staphylococcus aureus is found much more frequently in the nasal passages. About 50% of Americans have Staphylococcus aureus in the nasal passages. The lesion that you're most likely to see from Staphylococcus aureus is, what do they call that? A skin lesion. Sorry, I'm drawing a blank here. I'll have to think about it. I don't remember what that lesion is. Does anybody know? What's a skin lesion you get? Very common. Say again?
MRSA?
No, MRSA is a bacteria. It's a variant of Staphylococcus aureus that is resistant to multi-drugs, multi-antibiotics. It's a boil, that's what it is. It took me, I was saying, gosh, it's that lump on the skin, a boil. And then I said, oh, that's it, it's a boil. So if you ever get boils, then you might have yourself tested to see if you have Staphylococcus aureus. That's a normal part of your microbiota. Okay? Anyways, your normal microbiota can compete with bacteria that can be pathogenic. And how they can compete is they can prevent the growth of the pathogen by occupying the niche that the pathogen would occupy. And with our skin and our intestines, that happens all the time. That's why when you consume the wrong bacteria, one of them is unlikely to give you the disease. With Salmonella, you need at least 500, I think, Salmonella cells before you'll get Salmonella. With E. coli, and I'm talking about a variant that causes disease, not the normal E. coli variant that's a part of our normal microbiota, you need maybe hundreds of thousands of them before you get an infection. The reason is that our normal microbiota are already in the area, and they can occupy the niche and prevent the pathogen from occupying it, growing in that location, and then causing disease. Any question about that? The normal microbiota can compete for resources, keeping the pathogen from getting the resource so it won't grow. The normal microbiota can produce acids, making it so that the pathogen can't grow, it's too acidic. That happens with our skin and in our mouth and in the urethra. The normal microbiota make that environment more acidic, and that keeps many human pathogens from getting established because they don't like the lower pH. Does anyone remember what the pH of the skin is? It's something like around pH 4.
7.2?
No, it's not pH 2, that's your stomach. It's around pH 4, maybe 4 to 5, and it's your normal microbiota that makes it that acidic. And then the normal microbiota can produce bacteriocins that can prevent the growth of the pathogen. Most bacteriocins are proteins that the normal microbiota produces, and the bacteriocin acts like a very narrow spectrum antibiotic. Now, most narrow spectrum antibiotics only work on either gram positives or gram negatives. Like penicillin G, we consider that narrow spectrum antibiotic, and it works on most, if not, well, most, we'll just say most gram positive bacteria. Doesn't work on gram negative bacteria. The bacteriocin is much more specific. If the bacteriocin is produced by E. coli, it would only inhibit cells similar to E. coli, such as Salmonella and Shigella. It would not inhibit, like Staphylococcus epidermidis, which is totally different. What's another? It would not inhibit Pseudomonas, which is another gram-negative bacteria. Salmonella and Shigella are very similar to E. coli. Some people even say they're in the same family. Other people don't, but my point is they're similar to each other, and a bacteriocin that's produced by E. coli will prevent the growth of Salmonella and Shigella. And the bacteriocins, think of them as a very specific, very narrow antibiotic. I often wondered why we weren't using bacteriocins to fight an infection. And a few years ago, I read an article where somebody was experimenting with using bacteriocins to fight infections. So nowadays, they are being at least experimented with. But they'd be a very good antibiotic. If you had a salmonella infection, you produce the bacteriocin produced by E. coli, and then it'll prevent the salmonella. Any questions about microbial antagonism? All right, let's move on to probiotics. This is when we consume live microbes. You either consume them or you could apply it to the skin. And the purpose is to exert a beneficial effect. Like women who eat yogurt with live organisms in them are much less likely to get a yeast infection. And so probiotics can be, a yeast, what am I talking about? Yogurt could be a probiotic if it has live organisms in it. Other probiotics, you just take a pill that contains the organism. So the normal microbiota forms a relationship with their host, and that relationship we call symbiosis, where two different organisms or populations are living together. There are three different symbiotic relationships: the normal microbiota and the host. Their symbiotic relationships are commensalism or mutualism. Commensalism is where one organism benefits and the other is unaffected. For example, what's a good example? We'll say this one. It might not be entirely commensalistic because it can prevent the growth of a pathogen, in which case that's not commensalism, that's doing me good. But Staphylococcus epidermidis, most of the time when it's on my skin, it benefits by getting a home, getting nutrients that I produce for it. But at least some of the time, Staphylococcus epidermidis isn't doing me any good or any harm. So I'm unaffected. So that would be a commensalistic relationship. Okay? A mutualism is where both organisms benefit. An E. coli in my gut would form a mutualistic relationship. The E. coli gets a home and gets food to digest that I obviously have consumed. And then I benefit because E. coli helps me digest the food. So that's a mutualistic relationship. Any question about any of that? Now, parasitism is when one organism benefits at the expense of the other. So Staphylococcus aureus on my aunt was causing a skin infection. So Staphylococcus aureus was in a parasitic symbiotic relationship with my aunt. The skin was providing a home and nutrients for Staphylococcus aureus, and the Staphylococcus aureus was causing a skin lesion. All right, any question about any of that? So the normal microbiota do not normally cause disease, at least in healthy individuals. But sometimes the normal microbiota can cause disease. And if they cause disease, we tend to call it an opportunistic normal microbiota. An opportunistic microorganism is a bacteria, like the normal microbiota, that can become opportunistic pathogen and cause disease. Like normally, Staphylococcus epidermidis does not cause disease on my skin. And if it grows on my skin, everything is fine. But if I get a puncture wound, and then the Staphylococcus epidermidis is growing in the wound, that would be an opportunistic infection, and it would be causing disease. In which case, that Staphylococcus epidermidis would be opportunistic. Some normal microbiota can be opportunistic when the individual is compromised in their immune system, such as having AIDS, undergoing cancer therapy, or some other immunotherapy. And I gave an example with Staphylococcus epidermidis. I got this example with E. coli here. E. coli is normally found in the gut. If ever it's growing any place else on the human body, such as in the ear, such as under the skin, that would be an opportunistic pathogen. Oops, didn't mean to do that. Caused by E. coli. Staphylococcus aureus is normally found on the nose. In some patients, 20% of them found on the skin. If it causes a skin lesion, it's said to be an opportunistic pathogen. And some people call Staphylococcus aureus not opportunistic pathogen, but a frank pathogen, meaning a human pathogen. Depends who you're talking to. All right, any questions about how opportunistic microorganisms differ from the normal microbiota that normally do not cause disease? So So I suppose if your normal microbiota ever causes disease, then that one species is an opportunistic microorganism. And then when it's not causing disease, it's a part of your normal microbiota. All right, any questions about anything so far? If not, let's change gear a little bit and revisit Koch's postulate. And this is talking about the etiology, the cause of disease, the cause of infectious diseases. Remember, infectious diseases are caused by microorganisms, and that is why they are infectious. Robert Koch, you might remember, he's the one who showed that Bacillus anthracis causes the disease anthrax, and he was the first person to show that any organism caused a given disease. And with that, he literally proved the germ theory of disease, that microorganisms can cause disease. He then wrote up the Koch's postulates, the statements used to prove that a given infectious agent causes a specific infectious disease. So what you do with Koch's postulates is you have a sick individual, and then you isolate from that sick individual organisms that could potentially have created the sickness, the illness. So isolate the pathogen from a sick creature, and then you need to grow it up in the lab and obtain pure cultures. That's not shown, but what you do is streak it out for colony isolation 2 times, and then grow up that organism in vast numbers. And then you take that pure culture and you inject it into a healthy organism. And if that healthy organism then becomes sick with the same disease we saw before, we say that this organism causes that disease. And Koch's postulate goes one step further to prove that this disease is caused by this organism. You have to be able to isolate that organism from this animal, sick animal. Any questions about Bacillus anthracis and Koch's postulates? Okay, here's a question for you. Why is the bunny sometimes on its side and sometimes standing up? Why is the bunny sometimes on its side and sometimes standing up?
One's healthy and one is ill.
Yeah, the one that's standing up is the healthy bunny. The one that's laying down is the sick bunny. That's pretty obvious. That's what humans do, especially when they're really sick. Now you should know that there are exceptions to Koch's postulates. Like, what do we do if we can't culture the organism in tissue culture? There are microbes that we've never been able to successfully culture. Like Bartonella henselae, which causes cat scratch fever, syphilis. We've tried for over 100 years, it's probably over 120 years now, to be able to grow syphilis in tissue culture. And no one's ever figured out what syphilis requires from its host in order to grow. And so we've never been able to grow syphilis and some other organisms in tissue culture. And so obviously, you cannot perform Koch's postulates if you can't grow up the organism in culture. So that's an exception. We'll talk about how we know syphilis. used to know the name of the organism. I'm drawing a blank here, so I'll just talk about cat scratch fever. Why we know the cat scratch fever, the Bartonella henselae causes cat scratch fever. And it's not by using Koch's postulates, because we can't get this organism to grow in culture. Now, what do you do if multiple organisms cause the same disease, signs, and symptoms? Like with the human cold. It can be caused by 3 different families of viruses. And so you can't say that one virus causes the human cold. Some species can cause different disease conditions. Like, oh gosh, I just lost the name of that. Salmonellosis is the more gentle disease. And typhoid fever, I think, is the more severe disease. I might have that name wrong, but the point is that one organism in 2 different patients can cause 2 different diseases. In which case, Koch's postulates, by definition, don't apply. And then what do you do for moral and ethical conditions for human-only disease? Like, we think that HIV causes AIDS, but have we ever injected HIV into a patient to determine if they later get AIDS? No, we have not done that for moral and ethical reasons. Okay? So how do we know that the bacteria, which I'm still drawing a blank here on what the name of it is.
Or syphilis?
Yeah.
It is Treponema pallidum, and it was actually successfully cultured in 2018.
Oh, really? So I'm out of date here. Thank you. So they finally figured out how to culture it. They can culture it in rabbits. I'm talking about in a petri dish.
I haven't read that far into the article yet, but it sounds like there was some school that was able to culture it
In tissue culture. It's possible that they finally figured out how to culture it. But I gave the other example, Bartonella henselae, also, that they haven't figured out how to culture it. What do you do? So how do we know for HIV that it causes AIDS? How do we know Bartonella henselae causes cat scratch fever? We can't grow these organisms up and purify them and then inject them into a live organism. You couldn't do that for HIV because there would be moral and ethical reasons. And Bartonella henselae, we've never done it just because we can't get it to grow in culture.
I mean, don't they use like chimpanzees and stuff like that for that reason?
You can use chimpanzees. HIV does grow in one species of chimp. There are 2 species, I don't remember which one. But it doesn't cause AIDS when they get it to grow in the chimp. But they can get the virus to grow, so that wouldn't show that that causes AIDS. Because it doesn't cause AIDS in the chimp. Okay? No, there's another way, and that is we do vast epidemiological studies, and then we follow these patients over time. And someone who was not infected with HIV, and later they become infected, we follow them further in time, and then see what percentage of them become AIDS patients. And that's how we know that HIV causes AIDS. And that's how we know that Bartonella henselae causes cat scratch fever. And then with the viruses that cause the human colds, strictly speaking, you can't do Koch's postulates because one disease organism, one pathogen, is causing one specific disease. Well, we have many different viruses in 3 different viral families that are causing the same human disease. but we can grow up one species of virus and infect it into many patients and then see if they get the human cold. And that has been done. All right, any questions about any of this? A few more terms in classifying infectious diseases. A symptom is a change in the body function that is experienced by a person as a result of disease. It is a subjective term, a symptom, and it is not apparent to the observer. Like, for example, when I tell the doctor I've got a headache, there's no way the doctor can measure that headache and confirm that I have a headache. So the headache is a symptom. A sign, on the other hand, is a change in the body that can be measured or observed as a result of disease. Like when I have a temperature over 100 degrees Fahrenheit, that is a sign of disease. And anyone can measure my temperature. Any question about the difference between a symptom and a sign? Now, with that stated, this is the way the textbook uses these terms. You'll find that sign and symptom are used interchangeably by the public and by many doctors. I'm not going to say all doctors, but many of them. And generally speaking, I will use the terms interchangeably. So if I only say one, I mean both of them. A syndrome is a specific group of signs and symptoms that characterize a disease. Like the signs and symptoms characteristic of AIDS are the AIDS syndrome. You should know what a communicable disease is. That is any disease that can spread from one host to another. Measles, tuberculosis or TB, chickenpox are all examples of communicable diseases. A contagious disease is a disease that easily spreads from one host to another. So chickenpox and measles would be a contagious disease. So would the most common contagious diseases, colds and flus, very contagious. They easily spread from one host to another. What disease is communicable but not contagious? TB. It's communicable, it does spread from one host to another, but it doesn't spread easily. So TB would be a disease which is not contagious.
I got a question about TB for you.
Uh-huh.
I was heavily exposed to chronic TB as a child.
Uh-huh.
And why is that bump that show up in the arm, sir?
They just do a skin test to see if you've been exposed to TB, and then it'll react as a bump on your arm. Is that what you were talking about? They can do that test at any place on your body, but they frequently do it on the arm.
But does TB is transmitted like the Staphylococcus aureus, like by air or?
No, it's a bacteria. Let me think about how that one is transmitted. It can be obviously touch to touch, but I think TB, yes, it's definitely a respiratory disease too. And most people, when they get TB, they first get it growing in the lungs. And then if they spread it, they're likely to cough it out or sneeze it out, or maybe when they blow their nose, and then they touch like a doorknob, and then somebody else who touches the doorknob can pick it up. They would have to inoculate probably their mucous membrane, but they can pick it up. So TB is communicable, but it doesn't spread all that easy. So it is not contagious. So all these diseases that are contagious are also communicable, but only some diseases that are communicable are contagious. A non-communicable disease is one that does not normally spread from one host to another. For example, tetanus does not usually spread from one person to another. Tetanus you usually get as a puncture wound, and then you get tetanitis, a bad disease by the way, but so you get exposed from the environment, and then it can infect you, but it's unlikely to move from you to another person. In most cases it doesn't happen, but like cases of cannibalism, tetanus could spread. Okay, any questions about any of this? If there's no questions, I'm going to end it here. Come on. And we will start here next time. So I'll see you in the lab at 6:30.